So my talk is on insights on skinny old cats, and there was a lot of work went into finalizing this. And when they finally came up with this title, Insights on Skinny Old Cats, I sort of thought, well, what does insight mean? And insight means your ability to see the truth clearly. And that's a nice idea, but do we all see the same thing? And that reminded me of these sort of classic ambiguous images that you see. And this is one of the old ones. It's from Germany in the end of the 18th century. And I think it roughly translated, said, which animals are most the same? And the answer is rabbit and duck. And apparently that's a good illustration of German humor at that particular time. But it does make you think we don't all see the same thing when we look at the same thing. And that probably applies to my talk as well. So I'm going to give you my interpretation of what I think I see, and some of you may see different things, and that may lead to some interesting questions later on, hopefully. I'm going to start with some just simple observational data from aging cats that's been around for a while. I'm going to review some information from skinny old humans and just briefly go over frailty as opposed to sarcopenia, as opposed to cachexia, which other people will talk about later on. And then I'm going to talk a little bit about changes in body composition in aging cats, and then the role of enteric disease in the pathophysiology of why those body composition changes may be happening. I don't think it's a surprise. We all know that many middle-aged cats have problems with obesity, but when they get to be old, the major problem that cats have is that they lose weight. And that weight loss tends to be a harbinger of death before a year or two. It's not a good thing. It happens at different ages in different cats, but it tends to happen with all of them ultimately. And we're very familiar as veterinarians with things that commonly cause weight loss in old cats, and they're mostly very easily diagnosed. We have good tests for them now, and they're easily either ruled in or ruled out. The ones that get a little bit more difficult to deal with are chronic enteropathy, pancreatopathy, pancreatitis, and unexplained picky appetite, poor appetite. And that's still a bit of a challenging area. But you can rule out a whole lot of things pretty easily these days. And my interest in the old cat started back in the 1990s when the GI lab, which at that point was at Kansas State and then Purdue, we started measuring cobalamin in cats. And it became very immediately obvious to me that something was different between cats and dogs. Cats got a higher proportion of cats than dogs got low serum cobalamin, and the serum cobalamin levels in those cats was much lower than it was in dogs. The assays used to go really low to 29 back then. They were radium assays, and cats would often have undetectable levels. And it was actually pretty rare for dogs to have undetectable levels. So there was something different about cats. And this is actually data from a presentation at this meeting nearly 20 years ago now, just demonstrating that a lot of old cats get very, very low serum cobalamin levels, much more so than in dogs. And at that time, it was a little bit debatable whether that was of any significance because people didn't realize that nobody had proven that that caused any clinical consequences. And at that time, working with Craig Rouault at Texas A&M when I was at the GI lab, we developed an assay for methylmalonic acid and showed that that marker of cobalamin cellular deficiency actually goes up really high in cats with low serum cobalamins, and when you give them cobalamin, the methylmalonic acid decreases dramatically. And in that initial group of cats, we also showed that after simply 4 weeks of cobalamin replacement, which at that time we used to do by injection, those cats gained a significant amount of weight. 10 out of 19 gained weight. Now, not every cat with low cobalamin gains weight when you give it cobalamin, but some of them certainly do. So that really proved the fact that cats do get cobalamin deficiency, and it is important. This is some classic slides that Jim Morris from the University of California, Davis gave me years ago, showing the effect of feeding kittens a cobalamin-deficient diet. And they got these scruffy, wet-looking hair coats, which is interestingly the way that a lot of these old cats with weight loss look. They get scruffy, funny-looking hair coats. I think it's more than just cobalamin, but I think in some cases cobalamin contributes to that. And Dr. Morris showed in these cats, they really had very few other clinical signs, but they stopped eating. And then because they stopped eating, they stopped growing. And when they gave them cobalamin, put it back in the diet, the hair coat became normal, the cat started eating, and they became clinically normal. Another point I'd like to make is that cobalamin deficiency can have significant systemic effects. This is a slide that I love. It's from a paper back in 1978 taken from human beings with pernicious anemia who have selective inability to absorb cobalamin. And that's related to lack of intrinsic factor. And those human beings, if you look at the intestines, you find that they have a really severe enteritis. They have marked infiltration with inflammatory cells and huge villus blunting. And if you give those anemic people cobalamin replacement, the intestine becomes normal again without doing anything else. So in veterinary medicine, I think our cobalamin deficiencies are mostly related to intestinal problems in the first place. And I don't know how you'd prove it, but I'm sure that this goes on, especially in some of those cats with really severe cobalamin deficiencies. The intestine becomes even sicker because of the cobalamin deficiency. It would be nice to be able to prove that. I don't know how you practically can, but it's certainly a real thing in human beings. And it maybe shouldn't be that surprising because you need cobalamin to make DNA. And all cells that are growing and reproducing do that. So there are almost certainly pretty massive, often subclinical changes that can occur in the background with cobalamin deficiency. So what do we know a little bit based on what's been out there for a while? Cats with malabsorption have more frequent and more severe hypocobalaminemia than do dogs. Old evidence from the late 1990s, mostly from the Royal Veterinary College, showed that if you actually culture jejunal juice from dogs as opposed to cats, the cats have more jejunal obligate anaerobes than dogs based on classic bacteriological culture, not the fancy stuff that Dr. Sugodolski does now at the GI lab. This is old-fashioned stuff, but there were significant differences between dogs and cats. There's also some old evidence that I will touch upon later on that cats have much more permeable small intestines than do dogs. I think there's 3 different groups that worked on cat permeability quite a while ago now, but they all found that cats had leakier guts than dogs did. And we can summarize, I think, that cobalamin status in cats is certainly diagnostically and therapeutically important. And we also now know that parenteral and oral cobalamin, thanks to Linda Torreson's work, are both very effective at normalizing serum cobalamin levels. So it's a very treatable thing. While that work was sort of going on, Purina had been making observations in their colony of cats or colonies of cats that they use for digestibility studies and things like that, and had made the observation that many of the old cats developed a malabsorption. And they detected that by using classical digestibility studies. And these old cats, as age went on, more and more of them, up to about 30% when the cats became geriatric, had fat malabsorption. And they also, or fat maldigestion, let me say. They also showed that those same cats tended to get protein maldigestion as well. And I've looked at this figure for a long, long time, and it actually only is in the last working for this talk that something became apparent to me. It always seemed a little bit odd that these graphs don't go exactly in parallel. And I think there may be an explanation. Well, there is an explanation for that, and maybe I think I know what it is. We'll come back to that later on. But these old cats do get a progressive decline apparently in their digestive function with age. Other observations that they made, they did very careful observations, lots and lots of data. It's really quite a treasure trove of information, and it's hard to go through. But they also looked at the body composition using DEXA scanning, and they showed that, as might be expected, we'll hear about sarcopenia later on, cats do get sarcopenia. The lean body mass gradually goes down with age. And this is months from death at the bottom here. So it's a pretty linear loss of muscle mass with time. But interestingly, in these old cats, the cats that had the fat malabsorption had a different rate of body fat loss than did the cats that had normal fat absorptive capacity. So normal cats, their body fat content stayed pretty constant. And then in the last year of life, it started to go down. And once it started to go down, they died pretty soon after that. The cats that had poor fat absorption had a different rate of body fat loss. Starting about almost 4 years before they died, they gradually started to lose more and more body fat. And that's a little bit odd. And I think the explanation might be that they're not digesting and absorbing their fat in the food normally, so they start to basically metabolize their own body fat. And we have some metabolomic data that we published in EPI cats with pancreatic insufficiency, where there seem to be metabolic changes, which suggests that because the cats are not digesting their food properly, because they don't have the pancreatic enzymes, instead they're breaking down their body fat, and that leads to some interesting metabolic changes. So these cats, that they called them low-fat cats because they had low-fat digestibility, they start to lose weight earlier than the other cats, and then they tend to die within 4 years. Now, it makes it look like these cats all die at the same age, but in fact, this decline in the low-fat cats happened significantly sooner than in the cats that had normal fat digestibility. So they start to lose body fat, and then they decline, and that starts at an early age than the normal decline in cats with normal fat absorption. It's not just something that's been observed in colonies of cats that pet food companies use for their digestibility studies. People in a general practice setting have also noticed that just subjectively, when you're looking at older cats as part of a wellness examination, somewhere like 75% of the cats older than 15 have obvious muscle wasting. So it's a real thing, not just a pet food company observation thing. And just to summarize very briefly the differences between sarcopenia, cachexia, and frailty, we'll hear more about this later on. But sarcopenia is just something that happens with age. That's my simple summary of it. Cachexia happens when you have a significant other disease, like a cancer or heart failure or something else that's making you sick, and that leads to loss of muscle and fat. And frailty, though, is something where it's something in addition to sarcopenia, but you also lose fat in addition to lean body mass. And I think that fat loss in the low-fat cats that I showed you that starts earlier is probably a harbinger of frailty, because those cats are losing muscle and fat in the absence of apparently overt disease. In human medicine, people are aware of frailty, and they've tried to sort of make a specialty out of it and study it. There have been problems. There's no real good consensus on how to define frailty. And the papers that have been done, I don't know that any of them have made a huge impact on treatment of human beings or prolonging life. It's an interesting area. Frailty is a real thing, but it's difficult to work on. Frailty is probably multifactorial. There's a whole lot of things that are listed here that happen with increasing age. Interestingly, many of those types of changes we know happen in cats or any aging species. Things like chronic inflammation increase with age, and immunosenescence, and oxidative stress in particular. And there's some evidence that those things are somewhat important in the aging process. So it's not really sure exactly what the total explanation for frailty in humans is. And it's not difficult to diagnose in some cases. Sometimes it's painfully obvious, and you see it progressing rapidly, and it doesn't usually end well. And that's just a common sense type thing. But what about pre-frailty? Something's going on before people actually develop signs of frailty. And that's really the reason for doing frailty research, is to see, can you find something where you can intervene and maybe delay the onset of frailty, or perhaps even prevent it altogether. Examples of that would be eating a good diet, but also making up for some deficits. And the one that's perhaps most known about in people is that certain people tend to be vitamin D deficient just because of lack of exposure to sunlight and things like that, and maybe not eating the best diet. And so many, many people these days take supplemental vitamin D because they're not getting enough from the diet. And it is believed that may well, in fact, slow down onset of frailty, but I'm not sure that's actually been proven. But it's an example of a potential nutritional intervention where you can improve things because you've identified something before it leads to a problem. And another example would be getting some sort of regular exercise to help maintain muscle mass. It can also help slow down onset of frailty in human beings. In humans, they've had a variety of different scales to try and objectively assess frailty. They all suffer from the same problem, that many of the parameters are either self-reported or they're based on very subjective criteria. And that's not going to work very well in veterinary medicine. The American Association of Feline Practitioners has actually come up with pointing out that they believe that old cats do get frail. I think that's sort of common sense, some of them do. But how can you measure it? And can you even detect pre-frailty in cats? They point out that the reason for measuring frailty is that if you're frail and then you get another problem, you don't cope with the other problem just as well. And I think we saw an example of that in the COVID pandemic, where old and frail people just didn't do as well as people that weren't frail. And that's probably true in our cat and dog population as well, especially the cats, where they get old and thin and live for a surprisingly long period of time with multiple health problems, which can often be somewhat well managed. So avoiding frailty would be a good thing if we can pick it up. And the AAFP came up with some suggested starting points for evaluating frailty and even pre-frailty. And they talked about some phenotypic things like unintentional weight loss, weakness, early exhaustion, slowness, and low activity. And if you have all of those, you're definitely frail. And if you have two of them, you may be pre-frail and you should think about doing something. And they've also looked at some other frailty things that look at cognitive function. And it was really a set of suggestions, I think. And they were presented, adapted from human medicine, that maybe the feline practitioners might try and score some of these things and improve them over time. But it's early days. So to my knowledge, there's no published research on frailty in pet cats. There's a couple in dogs, which I'm not going to spend much time on, but one study, they showed that using a frailty index that they come up with, they could be moderately accurate in predicting short-term mortality. And another one which was done in guide dogs. Guide dogs tend to be very well observed. Now, most guide dogs retire long before they become frail, but they tend to be well looked after and well observed and have good medical records and things like that. And they could say that when there were more than 2 deficits in one of the categories that they looked at, those dogs were most likely to die in the follow-up period they looked at. So they were brave efforts, but I don't think they really were particularly helpful. And again, a lot of the observations were things that you can do in dogs. But when it comes to cats, we certainly see cats that have got arthritis and can't jump up on the counter anymore and things like that. But exhaustion, low activity level, those things are not, they're not easily routinely assessed in cats. Although I think there are some subjective feelings that if it's your cat, you know something's changing. But they're hard to score. They're hard to give a name to. This is a good example of English humor in 2022. So cats certainly are potentially capable of developing frailty as people do. They get a lot of the same age-associated pathophysiological changes, but we don't really have a good way to score them and monitor them. It would be nice to have some more objective measures. And again, we may hear something about this a little bit later on, but there is something that I find intriguing. In the last few years, a couple of papers talking about looking at the ultrasonographic evaluation of back fat thickness in cats at the level of the 13th rib, and also looking at muscle thickness in that same region. And I don't know whether anybody has taken this any further. I know they use that for food animals that we eat, to assess carcass quality and things like that. So it does seem a potential objective way to potentially look at the evolution of frailty in our cat population. So again, the goal of looking at frailty and thinking about it is to find out, can you find an intervention whereby you can reduce the rate of deterioration and stop animals becoming frail and hopefully prolong their lives with a good quality of life. So to go back to the Nestle Purina data about the low-fat cats, again, just to point out that there's something odd here. And the thing that's odd is, it looks like 12 to 14 years of age, 14 years of age, the fat digestibility is the same. But somewhere between 12 to 14 and 14 years of age, the protein digestibility actually becomes more than twice as bad. So there's a discrepancy there. You'd think if the intestine was just becoming dysfunctional and not working, that those things would sort of change in parallel, and they didn't. And so that's one observation that we will come back to. Something's going on there. These low-fat digestibility cats, just to remind you, those that are not familiar with it, they do not usually have diarrhea. The stool probably does have, well, it does have more fat in it, but it's usually not that obvious. This is a low-fat cat that's got totally normal stools as far as I'm concerned. And that's really quite common. They do not, by any means, all have diarrhea. The condition with the low-fat malabsorption is associated with low vitamin E levels, low vitamin B12 levels, low folate levels in some cases, changes in thin skin thickness, body fat, and body condition score. And these observations have been duplicated or remade in other academic environments away from pet food companies, where a 9% decrease in fat digestibility was noted in cats that had subnormal cobalamin levels compared to those with normal serum cobalamin. So these associations appear to be real and in different parts of the world as well. So serum tocopherol and cobalamin have strong inverse associations with digestive function. These are easy tests to perform. People don't do fecal fat levels anymore. It's not something you can get done in practice, even if you wanted to. But it is easy to do a serum vitamin B12 level or a serum vitamin E if you send it to the right diagnostic lab. The other one that's been reported is that cats with chronic enteropathies have low 25-hydroxyvitamin D. And I have no idea what that means, but it does, apparently it's a fairly constant thing in idiopathic chronic enteropathies, inflammatory enteropathies, and intestinal small cell lymphoma. I think, and I'm not a nutritionist, but I think there's significant doubt as to whether 25-hydroxyvitamin D is actually a really good indicator of deficiency, but something is different with regard to vitamin D in these cats with chronic enteropathies. Okay, so we're going to move on to some new things. Just to recap, though, cats with malabsorption have more severe and more frequent cobalamin malabsorption or hypocobalaminnemia than do dogs. Cats apparently, at least on culture, have more jejunal obligate anaerobes than dogs, based on culture. Their intestines are more permeable than dogs. And cats that have abnormal cobalamins and folates have other changes as well, which Jan alluded to in his previous lecture, related to bile acids and tryptophan. And these things must indicate there's a dysbiosis. And in the next few slides, that's what I'm going to talk about. Just to make a point, I do a lot of consults with veterinarians from the GI lab or through the GI lab. I still do consults on that side of things. And a lot of veterinarians think that, oh, if you don't have a high folate, then there's not dysbiosis, or there can't be dysbiosis if the cobalamin's not low, or you have to have both a low cobalamin and a high folate to have dysbiosis. Just to make it clear, you can see low cobalamin and high folate with dysbiosis. It used to be called small intestinal bacterial overgrowth, and that was originally how it was discovered in people in the 1970s. But you can see either or both or neither of those things with dysbiosis. So these are proxy markers for dysbiosis, but they're not always abnormal at all. So just be aware of that. They're useful to do clinically, but they're not tight associations with dysbiosis. Cobalamin probably is strongly related to what's going on with the microbiome, and this is some of the work that I did with Nestle Purina quite a while ago now, and techniques are changing, and there's other ways to look at this. But this did indicate that we could actually look at the spectrum of bugs that we could find in fecal samples and actually predict mathematically from the bugs that were there what the serum cobalamins would likely be. And the microbiologists that we worked with on this did a lot of studies on the types of bacteria that were grown in these different cats. And the bottom line was that cats that had normal to high cobalamins often had relatively low numbers of bacteria that are known to consume cobalamin, which is most bacteria. But some of them even had types of bacteria that could participate in cobalamin synthesis. Whereas the cats that had very low cobalamin levels, they had large numbers of bacteria that could not synthesize cobalamin and were almost certainly just using it up. So there was a relationship between what bugs were there and what the serum cobalamin levels were. And we move on to a little bit about conjugated and unconjugated bile acids. Jan talked a lot about primary and secondary bile acids, but another aspect of bile acid metabolism is conjugation or lack thereof. This just is a plain, simple bile acid. But the bile acid that the liver makes and secretes are conjugated with taurine, as I'm sure you all know. And that's very, very important. The other thing that happens is that bile acids get converted from primary bile acids to secondary bile acids. And that is just by chopping off a hydroxyl group, which, as Jan pointed out, is done by seemingly a very specific type of bacterium. So there's conjugated and unconjugated bile acids, and there's primary and secondary bile acids. In normal circumstances, the bile acids are conjugated with taurine. They're secreted in bile that way. They get secreted into the gut. They stay in the gut because they're conjugated, and they participate in fat absorption. And then in the ileum, they're reabsorbed and then recirculated through the liver. So they facilitate fat absorption. Now, one of the things that can happen is that some bacteria, many bacteria actually, will deconjugate the bacteria. They will chop off the taurine, and that leaves an unconjugated bile acid. Unconjugated bile acids don't stay in the gut lumen. They passively diffuse into the bloodstream very quickly. And when they leave the gut, they're no help in fat malabsorption, which is why bacterial deconjugation can contribute to steatorrhea. But the other point is you get abnormal amounts of unconjugated bile acids in the bloodstream. And as Jan pointed out, those bile acids in the bloodstream bind to receptors around the body and have a whole bunch of systemic effects. And different primary and secondary bile acids and unconjugated as opposed to conjugated bile acids have different types of interactions with those receptors scattered around the body, which is why you can get systemic metabolic abnormalities associated with these changes in bile acid metabolism. And we did look at some unconjugated bile acids quite a few years ago in serum when Craig Rouault was working with us at the GI lab. And we found some interesting results. And this is just serum unconjugated bile acids in healthy cats. In healthy cats, they're very, very low. Nearly all of the bile acids are conjugated. But in cats that have GI disease as manifested by a low cobalamin level, the unconjugated bile acids in serum are much, much higher. So that must be bacteria that are making that happen. And now we'll move on to secondary bile acids, primary and secondary bile acids. And at the time that we did this work in serum, we had no idea what it meant. We could not explain it. Now, thanks to Jan's work and filling in a lot of the gaps, we can make some sense out of it. But way back when, we looked at serum circulating bile acids. Control animals had very, very low levels of primary bile acids, but higher levels of secondary bile acids. Surprisingly, the cats that we knew had enteropathies with low cobalamins, they had much higher levels of primary bile acids than secondary bile acids, which fits in very nicely with the things that Jan told you about Hieronymus and the bugs that convert primary to secondary bile acids. So clearly there's very, very strange things going on in these cats with regards to primary and secondary bile acids and conjugated and unconjugated bile acids. And that doesn't just have effects in the gut. As Jan pointed out, Those abnormal bile acids or different bile acids circulating around in the bloodstream have distant metabolic and complicated effects which are only just beginning to be understood. Another aspect of cobalamin is that I think at the same time that Linda Torreson was doing her work on oral administration of cobalamin, we did a little study in cats in the colony that we knew had this chronic enteropathy, giving them oral cobalamin in very high doses. And we started out with crazy high doses. And the interesting thing was that the blood levels did go up very rapidly. They went up very, very high. And we were giving like 1 or half a milligram a day. I think 100 micrograms is a much more rational dose, and actually the cats only need like half a microgram a day to be normal. But anyway, we gave these cats cobalamin, and then after 2 months, we stopped giving them the cobalamin, and we watched how the cobalamin went down again when we stopped giving the cobalamin. And it was quite interesting. We found that we could divide the cats into 2 groups. After 1 month, one cat's cobalamin had actually become undetectable. And it was way, way above normal a month previously. Kenny Simpson had previously documented in some cats at Cornell that the half-life of cobalamin in cats with enteritis was much shorter than the half-life of cobalamin in healthy cats. Well, in our group of cats, we found that there were 5 cats that after 3 months, their cobalamin levels had become subnormal. And then there were another group of cats where the cobalamin levels had stayed within the reference range. So we called those good responders and poor responders. And the poor responders were the ones when you stopped giving them cobalamin, the cobalamin went down again. And we looked at the microbiome in those cats, and it was amazing. There was a huge difference when you did principal component analysis between the poor responders and the good responders. Clearly, the poor responders had a very different microbiome from the good responders. And I think that illustrates one of the things that once these cats go wrong, they're probably stuck with a wrong microbiome for a long time. And these diseases, this change in age is not going to go away. It's something you kind of have to manage. So my recommendation has always been that you should start cobalamin supplementation when the cobalamin gets to less than around 300. And once you start it, you should plan on doing it for life, because the underlying problem is not going to go away. The microbiome is probably going to be abnormal for a long time, if not forever. So I'm now going to move on to alpha-1 proteinase inhibitor, which probably, I think most people know nothing about that protein. It's really kind of very interesting protein. Jan knows about it and hates it because he spent quite a lot of time trying to purify it at the GI lab years ago, which is a soul-destroying job. It's not an easy protein to work with, but it's interesting. And we've got some data on these old cats that I think is interesting to talk about in the context of stuff I've just presented. So alpha-1 proteinase inhibitor is the second most common protein in the bloodstream after albumin. And it's not just a protein, it's a glycoprotein, so it's got a lot of carbohydrate residues about it. And it has a sort of a business end here, which sort of sticks out. And as its name suggests, it's a proteinase inhibitor. Its other name is alpha-1 antitrypsin, or alpha-1 anti-neutrophil elastase. It's a very interesting protein, but we don't need to go into all of that. Bottom line is it's a proteinase inhibitor, and it has a bit on the end here that sticks out and makes it do what it does. And if you think about albumin and alpha-1 proteinase inhibitor, they're about the same size. But when albumin leaks into the gut, it gets broken down by the digestive proteases, and it gets chopped up into little peptides that are non-immunoreactive. So if you're losing albumin into your GI tract, you can't measure it in the feces because it's been digested. Alpha-1 proteinase inhibitor is not like that. When it gets lost into the gut lumen, it binds with the digestive proteases and it inhibits its own digestion. So it comes out in the poop as a complex with alpha-1 it comes out in the poop as a complex which is stable and can be measured by an immunoassay. And that's the basis of the alpha-1 proteinase inhibitor assay, which is available from the GI lab for dogs. It was available for cats. It's not anymore. But the basis is that if you've got protein-losing enteropathy, you've got holes in the intestine, protein leaks, and the albumin, the green stuff, gets digested into fragments, but the alpha-1-PI doesn't. It comes out intact in feces. And the only way, if a cat has alpha-1-PI in its feces, it's either losing its own protein or it's eating other cats, because there's really no other way that it can get there. Catherine Fetz, now Burke, after she got married, did these studies in setting up this assay and showed that this test was more sensitive for chronic enteropathy and neoplasia than measuring serum cobalamin, for example. There was no correlation with serum albumin, so these cats could have protein-losing enteropathy, but they maintained a serum albumin. Well, we applied this test to some of the cats with this old-age enteropathy, and it was quite striking. 11 out of the 15 cats that we looked at had high fecal alpha-1-PI, quite dramatic. Compared with the cobalamin, only 4 had abnormally low cobalamin levels in those 15 cats. So it seemed to be a pretty sensitive test. These cats apparently have protein-losing enteropathy. They don't have panhyperproteinemia. They don't even have hypoalbuminemia, but they are losing protein into the gut lumen. So alpha-1-PI is abnormal in many cats with malabsorption due to idiopathic chronic enteropathy, even when all the other tests are normal. So I would suggest that enteric protein loss probably contributes to the weight loss in these cats. The other interesting thing is that the presence of endogenous proteins in the gut might be interpreted falsely as poor digestibility. Because when Nestle and other people do these digestibility studies, I'm pretty sure that they measure nitrogen in the food and nitrogen in the poop. And the assumption is that is reflecting digestion. But if you're losing your own tissue proteins, that's also going to be nitrogen in the feces. And so I wonder whether this discrepancy between the fat and the protein reflects that these really old cats start to lose a lot of protein into the gut, and it can be misinterpreted as low protein digestibility, but it's impaired protein digestibility plus enteric protein loss. Now I'd like to move on to tryptophan. Jan mentioned this. It's a very topical thing in the microbiome enteropathy world at the moment. And he mentioned the fact that microbial metabolism of tryptophan leads to the production of indoles. And these microbial indole catabolites of tryptophan are thought to be very, very important. Again, just to summarize what Jan said, they apparently have effects in the local enterocyte population. They bind to certain receptors. This is tryptophan. These are some of the indoles. There's a whole bunch of them. And these indoles are only made by bacteria. They're not mammalian. But they have an effect on mammalian cells. They bind to receptors, and they end up actually modulating, decreasing inflammation, and they also increase the production of tight junction proteins, so they make the tight junctions tighter, and they also stimulate mucin production. So they have beneficial effects, and you need these microbial indole metabolites to have a normally functioning gut. And we measured these recently. This is data from literally two weeks ago, done in collaboration with colleagues at the GI Lab and Cinna Marcelio at UC Davis. And we set up the indole assays and other assays at Illinois. And these are the results we got briefly to try and make it as simple as possible. These were old cats, healthy older cats, some with chronic intestinal inflammatory enteropathy, and some with small cell lymphoma. They all had lower than normal body condition scores. Their serum cobalamins were low, just like so many of the skinny old cats that we talk about. And interestingly, they had significantly increased TLI and PLI, which is another story. What is this link between the pancreas and the skinny old cats? There's a link there that I don't have time to talk about. But they were skinny cats with poor body condition score, one of two different categories of enteropathy, and low serum cobalamins. The tryptophan levels in the serum of these cats was decreased compared to the controls. The ratio of tryptophan to kynurenine that Jan talked about, it's one of the ways that you look at this intestinal aspect of tryptophan metabolism. They were significantly elevated in the cats with small cell lymphoma and inflammatory enteropathy. And the microbial indole catabolites of tryptophan were also low in the affected cats compared to the healthy cats. And all of those things correlated with cobalamin in those same cats. So again, there's something microbial going on that's affecting indoles, it's affecting cobalamin, it's affecting bile acids. It's sort of a recurring theme of interrelated things, again, emphasizing what Jan said, that everything seems to be very, very interrelated. And so one interesting aspect of that is that if you don't have these indole catabolites, you're probably not going to maintain your tight junctions properly. That is a feature of all the experimental studies that we've read about animal models of tight junction and tryptophan metabolism. So probably when the microbial indole catabolites go down, you get decreased mucin, you have less tight junction protein, and maybe increased inflammation too, all as a consequence of that. And if you have leaky tight junctions don't just keep things in the gut lumen, they also keep things in the body. If you've got big leaky tight junctions, you may well expect that I would think you might be losing albumin and other proteins across those tight junctions. So microbial effects can have good and bad effects. Jan's talked about short-chain fatty acids from carbohydrates, changes in bile acid metabolism that affect their function in the gut, and also the systemic metabolic effects, and then the metabolism of tryptophan to indole catabolites that might have changes on intestinal permeability and inflammation. There's evidence for all of these things playing a role in cats. To go back to something a little bit more immediately practical, Purina did a study starting back in year 2000. It's all written up in the literature by Carolyn Cupp and Avi Patel and others who don't work with the company anymore. But they compared the effects of feeding one of 3 different diets to cats that were at least 7 years of age, and feeding that diet indefinitely for the rest of the cat's life. And then they did digestibility studies and monitored the body composition and just observed what happened. And they found that the low-fat cat thing, the weight loss and the poor fat digestibility happened in each of the 3 groups of cats fed these experimental diets. But in one of the 3 groups, the cats developed the enteropathy later, about over 12 months later. And they also started to lose weight later, and they lived longer, which was kind of remarkable. And the one thing that was very different about that diet is that that diet contained whole dried chicory root as a prebiotic source. It also had alpha-tocopherol and beta-carotene and fatty acids in it as well, and one of the other diets did as well. But the diet where they did really well had the antioxidants as well as the fatty acids and the dried chicory root as a prebiotic. What it was that made a difference, I don't know, but the evidence looked pretty strong. They lived about a year longer, more than a year longer than the other cats, and they showed a proportional delay in the onset of abnormalities associated with this enteropathy. And I wonder whether it would never been presented that way, but it's almost like that diet had an effect on pre-frailty, because those cats did become frail. They just became frail later. So I think it was maybe a serendipitous finding, but it probably was some effect on pre-frailty before frailty became apparent. So like the difference in lifespan was about a year. Oh, it was over a year between the basal diet and the most supplemented extra diet with the prebiotic in it. So you may be able to prevent some of this, or at least delay it, but what do you do when it's too late, which is when we usually get the consults as veterinarians? Well, you can certainly, we don't give antibiotics. I don't think I need to say any more about that. It's hard to get those antibiotics into cats anyway. They hate tylosin, they hate metronidazole. I don't know why veterinarians feel obliged to try and do that, but it's time to stop. Correct abnormalities. Certainly cobalamin and folate if it's abnormal and can be corrected. Tocopherol has kind of been a little bit neglected, but I think I don't think it makes cats immediately very sick, but I think some cats probably might benefit from tocopherol supplementation because I know some of them are deficient in spite of being on good diets. Should we do anything about vitamin D? I don't know. When it comes to dietary change, my own personal preference, and it's totally testimonial, anecdotal evidence, is that the cats that have chronic diarrhea, I like to treat them with a low-carbohydrate diet because I think that helps with the diarrhea. I don't know whether it helps with other aspects of what's going wrong with them, but it does seem to help with the poop, which is often people's immediate problem. I have no idea whether hydrolyzed diets is better for these old cats. Maybe somebody else has some feelings about that. It would seem logical to put a prebiotic in there if there's not one there. And many of the Purina products contain inulin, which is an extract of chicory root, as opposed to chicory root. And with that, the senior cat study that I told you about, what is it in the chicory root that might have made a difference? Is it just the inulin, or is there something else in there as well? And I think the easiest way to do these things is to actually feed a diet like the ones that have been modeled on that senior cat diet that contained the chicory root. It seems to be preventative. I don't know whether it has benefits in the cats that are already sick. I just don't know. Probiotics, I wish they work, but I don't know whether they do or not. Something that people forget about that I always like to mention when I give a talk like this is prednisolone. Everybody thinks about prednisolone as being anti-inflammatory and immunosuppressive, but prednisolone actually switches on genes and protein synthesis. And I think it does that in the gut. I know it does that in the gut, just like it does in other organs. And so if you've got a gut that's not completely normal, when you bathe it in prednisolone, it switches on genes, it switches on protein synthesis. It probably makes more proteins, including perhaps tight junction proteins, and digestive proteases and disaccharidases in the brush border membrane, and carrier molecules, probably all switched on by prednisolone. I have a doubt whether it's really an anti-inflammatory effect or an immunosuppressive effect. I think it's probably a boosting function in general effect. And I really don't have any feeling about chlorambucil and cyclosporin. I think of them as last-ditch efforts in these types of cats, but I'm interested in other people's opinions about that. So to try and summarize this, skinny old cats may be frail in a scientific meaning of that term, but we need more practical studies using objective measures. Maybe those ultrasonic evaluations of muscle and fat may be helpful. There may be metabolic markers that are to be determined that may be helpful. But it would be nice to have some objective measures to say that this cat is heading south, we need to do something now. Many of these cats do have a chronic enteropathy associated with dysbiosis. I think those are things that are worth looking for in these frail, failing old cats. Affected cats that do have the enteropathy commonly have enteric protein loss, and many of them also have chronic pancreatitis as well, which is really interesting, and it's another talk, what's going on there. But that protein-losing enteropathy is probably exacerbating poor digestibility and malabsorption of selected nutrients. Based on the senior cat study, there's already evidence that nutritional intervention may delay the development of enteropathy-associated frailty, and it may improve health and prolong life. And it would be nice to sort of have some additional objective evidence about that in a pet population, but it's difficult to impossible to sort of do those studies. So the mechanisms of these effects remain to be determined, but something's going on when you feed cats that special diet, and they live over a year longer than cats in the control groups. Hard to ignore that. Once you've got the problem, therapy basically needs to be multifaceted and individualized, and probably be given for life. Just like if they malabsorb cobalamin, they're probably going to do that forever. If you stop giving cobalamin, you're just waiting for an insidious deficiency to come back. So once you start therapy, you don't cure this disease at this point. You basically manage it like a lot of chronic enteropathies. We can intervene to treat some things. Cobalamin and tocopherol are logical, safe things that you can do. I've put taurine there in brackets just to remind us all that the whole taurine study was, at the end of the day, explained away by an effect of dysbiosis. So it's been there a long time, and it's definitely there now. Vitamin D, I don't know. We've got some evidence that tryptophan plays a role. Is supplementation beneficial? We don't know. It might be. It would be logical to look at it a little bit more. I showed serum levels of tryptophan. Don't forget that the actual indole metabolites are working in the gut. And while the serum effects looked at some level relatively subtle, they were completely consistent. But they were also serum, not what's happening in the gut. But what happens in the serum is probably reflecting what's going on in the gut. So feline GI microbiome dysbiosis may affect carbohydrate production and short-chain fatty acid production, cobalamin homeostasis, bile acid homeostasis, and tryptophan and indoles. Those things are all going on in these old cats. Enteric protein loss likely contributes to loss of body condition, and there are striking associations between this idiopathic chronic enteropathy and chronic pancreatitis, which are worthy of further study. And finally, I think there are prospects for positive interventions to delay the onset of frailty. And thank you for your attention.